Our first presentation is by the chair of the IFCC Task Force on COVID-19, who is Dr. Giuseppe Lippi from Italy. Dr. Lippi is a full professor at the University of Verona and he's a clinician scientist, and graduated in 1992 in medicine and is specialized in biochemistry and lab medicine. He has extensive experience in the area of clinical chemistry research and has published extensively in this area. And he's, as I mentioned, the chair of the Task Force on COVID-19 for IFCC. He's also the secretary of the EFLM, the European Federation of Laboratory Medicine. So he's going to present to us on molecular diagnostic testing of COVID-19, basically the testing of the viral RNA. Dr. Lippi, please.
Thank you, Koshav. Dear friends and colleagues, first of all, let me thank you, Professor Adeli, and the entire IFCC Executive Board for inviting me to chair the IFCC Task Force on COVID-19 and for inviting me to deliver this webinar on behalf of the IFCC, which will be focused on the recent IFCC guidelines that we have developed for SARS-CoV-2 molecular diagnostics. Before specifically addressing the main technical aspects of molecular diagnostic testing of SARS-CoV-2 infection, let me briefly introduce some aspects of this novel pandemic disease. Coronavirus diseases are not really new for humanity, since COVID-19 is the third coronavirus outbreak that has occurred over the past 20 years, following several acute respiratory syndrome in 2002-2003 and Middle East respiratory syndrome in 2012. The most notable difference is that the two former outbreaks have caused altogether less than 2,000 deaths, while COVID-19 has already affected over 31 million people worldwide, causing nearly 1 million deaths. This is still far from the over 50 million casualties that have occurred during the Spanish flu pandemic in 1918 and 1919, but it has been calculated that the COVID-19 outbreak would have caused nearly the same number of deaths if appropriate restrictive measures would not have been undertaken all around the world. SARS-CoV-2, as all other coronaviruses, is an envelope virus with positive sense single-strand RNA genome, encoding 4 leading structural proteins, which are known as spike, and which also contains the receptor binding domain through which the virus binds to its natural receptor at the surface of the host cells, the envelope protein, the membrane protein, and the nucleocapsid protein. The genome of SARS-CoV-2 also contains additional genes which encode accessory proteins, including the RNA-dependent RNA polymerase, which is essential for viral replication. The tomographic slice of SARS-CoV-2 virions are shown in the upper part of this figure, with the spike protein shown at 5 different angles on viral surface. Overall, SARS-CoV-2 has a mean size of 91 nanometers and displays a mean number of 24 spike proteins at its surface. This scanning higher magnification image clearly shows the structure and density of SARS-CoV-2 viral particles generated by infected human airway epithelial cells and released externally, ready to infect many other cells. After this succinct biological introduction, we can now concentrate on the molecular diagnosis of COVID-19. According to the World Health Organization, a confirmed COVID-19 case is a person with laboratory diagnosis of COVID-19 infection, independently from the presence of clinical signs and symptoms, and which thus places laboratory diagnostics at the very core of this pathology. The routine diagnostic confirmation shall then be based on detecting unique sequences of virus RNA by nucleic acid amplification tests, such as RT-PCR, while nucleic acid sequencing can be used for confirmation only when this will become necessary. Although the molecular assay originally developed and validated by the World Health Organization has been constructed employing primers and probes for amplifying the E gene and the RNA-dependent RNA polymerase gene, along with a confirmatory assay based on amplification of the N gene, the organization now declares that whatever among the N, E, S, and RNA-dependent RNA polymerase genes can be targeted. The use of 2 different gene targets is, however, warmly recommended. The World Health Organization has also endorsed some technical and clinical recommendations for diagnosing ongoing SARS-CoV-2 infection by means of molecular testing, whose main aspects are summarized in this table. Basically, the assay shall target at least one specific SARS-CoV-2 gene. The analytical sensitivity shall be equal or lower than 1,000 genomic copies per ml in respiratory tract specimens, but preferably equal or lower than 100 genomic copies per ml in upper and lower respiratory tract specimens and stool. The analytical specificity shall enable to detect only circulating SARS-CoV-2 viral strains. The diagnostic sensitivity shall be equal or greater than 95%, but preferably equal or greater than 98%. The diagnostic specificity shall be equal or greater than 99%. The analysis shall be at least qualitative, but preferably shall be quantitative. The turnaround time shall be at least lower than 4 hours, but preferably lower than 45 minutes. The throughput shall be of at least 50 to 150 samples in 4 hours, or up to 4 samples in 45 minutes, but preferably 200, 500 samples in 4 hours, or up to 6 samples in 45 minutes. And finally, the quality assessment shall include at least positive and negative controls. RNA extraction check might also be advisable. The CDC has also developed its own RT-PCR assay, whose primers and probes are designed for specific detection of the N gene, while an additional primer probe set can be used to amplify the human RNase P gene. In keeping with this concept, the key clinical indication of SARS-CoV-2 molecular testing, according to the guidelines of the International Federation of Clinical Chemistry and Laboratory Medicine, entails diagnosis of an acute viral infection during symptomatic illnesses, typically in the interval between onset of symptoms and the following 2 weeks. Support for clinical assessment of a symptomatic, pre-symptomatic, or mildly symptomatic patient for whom exposure to COVID-19 positive cases has been detected. Support for screening of asymptomatic, pre-symptomatic, or mildly symptomatic individuals in various contexts, which include but are not limited to prior to scheduled surgery or delivery, travels, hospital discharge, return to work or to school, as well as for managing small local outbreaks. Before specifically addressing the characteristic of molecular testing for diagnosing SARS-CoV-2 infections, I would like to introduce you to a widely accepted definition of NAAT, which stands for Nucleic Acid Amplification Test, and that is conventionally defined as an analytical procedure used for detecting specific nucleic acid sequences of a given species or subspecies of microorganism in a vast array of sample matrices. These techniques may include, though are not limited to, polymerase chain reaction based on nested, quantitative, or real-time reverse transcription, along with loop-mediated isothermal amplification techniques and quantitative nucleic acid sequence-based amplification methods. Another IFCC recommendation is then focused on identifying specific parts of population that should be prioritized for molecular testing, classified according to resource availability. Therefore, in resource-limited environments, molecular testing shall be prioritized in all those individuals presenting with acute respiratory illness, especially with fever and at least one sign or symptom of respiratory disease, such as cough or shortness of breath, as well as in all those subjects who have been in strict contact with confirmed or probable COVID-19 cases during the past 2 weeks. Then, molecular testing shall also be prioritized in groups and settings at higher risk of infection, such as older patients or those with pre-existing comorbidities that may include cardiovascular disease, diabetes, cancer, and so forth. The next IFCC recommendation highlights 3 important aspects in molecular testing for SARS-CoV-2. Basically, all laboratory services should implement an appropriate assay according to their desired clinical application, for example, for screening, diagnosing, or monitoring COVID-19, considering also that the diagnostic performance of many current point-of-care tests have not been well demonstrated so far. As concerns the viral gene target selection, some interim consideration can be made, considering also the current indication of the World Health Organization and CDC. Briefly, insufficient evidence still exists to suggest the definitive advantage of selecting molecular assays targeting a specific SARS-CoV-2 target. Nevertheless, it seems quite reasonable to recommend that these assays should be based on not less than 2 gene targets, preferably employing one primer capable to detect several coronaviruses, obviously including SARS-CoV-2, combined with a second set of primers which shall more precisely detect one specific SARS-CoV-2 gene. A brief description of the most widely used RT-PCR tests is summarized in this slide, which emphasizes the considerably different assay condition, starting with the use of different molecular gene markers across the different validated assays. As concerns the reference specimens to be collected, these are respiratory materials and include either upper respiratory tract specimen, represented by nasopharyngeal and oropharyngeal swabs, or lower respiratory specimen, represented by sputum and or endotracheal aspirate or bronchoalveolar lavage, especially in patients with severe respiratory disease. The IFCC guidelines substantially reflect those of the World Health Organization, but introduce some other important concepts. In summary, the settable type of specimen should always fulfill the recommendation of the manufacturers. An upper respiratory tract specimen shall be collected at minimum. More evidence would be currently needed before upper respiratory specimen could be replaced by saliva. Self-collection kits cannot be currently recommended unless appropriate instruction or patient education is deliberate. In such case, the test results of self-collected specimen shall also be interpreted with great caution. Finally, the practice of specimen pooling shall only be used in low prevalence or low resource settings, preceded by appropriate validation of the entire procedure. Although upper respiratory tract specimens still remain the preferred sample for detection of SARS-CoV-2 RNA, it seems important to mention here that the diagnostic accuracy, especially the diagnostic sensitivity of RT-PCR in these specimens, appears far from optimal. Cumulatively, the sensitivity in no specimen is between 50–60%, while that in oropharyngeal specimen is even lower, between 30–40%. Combining nasal and oropharyngeal specimens, the final sensitivity would range between 70 and 80%. The low diagnostic sensitivity of RT-PCR in upper respiratory tract specimen is attributable to a myriad of pre-analytical and analytical problems related to sample collection and analysis, which is summarized in this CCLN paper, but it's also due to the biological characteristic of the virus. In fact, as you will see in this figure, during the first days of infection, when the patient is incidentally still pre-symptomatic, the viral load in upper respiratory tract specimen may still be lower than the limit of detection of the molecular assay. The same situation may occur at the end of the infection, for a period that can last for 1 to 2 weeks when viral shedding at lower intensity is still present, but cannot be detected with many molecular biology methods, especially directed ones. In both these two windows, there is a high risk of generating false negative results when the diagnosis will only be based on nasal and oropharyngeal RT-PCR testing. In this interesting study, for example, Zhang and colleagues reported that the sensitivity of RT-PCR for detecting SARS-CoV-2 in nasal and oropharyngeal specimens is directly related to the number of samples collected and tested, increasing from 86% with one single test to 93% with 2 consecutive tests, and finally reaching a value of 100% only after the 6th consecutive test. It is also important to mention here that many pre-analytical and analytical factors may have an impact on the diagnostic sensitivity of molecular testing. which include, for example, identification errors, inadequate procedures used for specimen collection, handling, transport, and storage, collection of inadequate material for quality or volume, presence of interfering substances, manual errors, specimen contamination, or even the presence of interfering substances such as antiretroviral therapy. As concerned the potential analytical error, this may include tests performed outside the diagnostic window, active viral recombination, which may then impair probes annealing, use of non-sufficiently validated assay, lack of harmonization of primer and probes, which would hence provide non-comparable results among different laboratories, general instrument malfunctioning, insufficient or inadequate material for testing, non-specific PCR annealing, misinterpretation of expression profile, and use of non-clinically validated diagnostic cutoffs. Interesting data are also emerging on the potential use of saliva instead of the reference naso- and oropharyngeal swab specimen. In this study, for example, the authors not only found that the overall RNA viral detection rate was 10% higher in saliva than in nasopharyngeal swabs, but SARS-CoV-2 titers in patients with COVID-19 may also be higher in saliva than in nasopharyngeal swabs, so that this sample metrics may yield better detection sensitivity throughout the course of infection. Sample pooling is another interesting opportunity that has recently been addressed for purpose of enhancing the efficacy of molecular testing. Briefly, the strategy encompasses the process of pooling together and then screening a variable number of clinical specimens, while individual sample will only be assayed when the pool generates a positive test results. Some published reports have recently highlighted that under specific conditions, this strategy may be feasible for counteracting local shortage of reagents or limited test availability in intensive testing areas, for enhancing the throughput and reducing the turnaround time, as well for supporting cost-saving policies. Sheriff and colleagues in this study calculated that the optimal strategy would encompass the creation of pools with an optimal size of 5 to 13 individual clinical specimens in a real-life scenario. Where prevalence of COVID-19 would be comprised between 1 and 10%, the RT-PCR on oropharyngeal and nasopharyngeal swabs has a detection limit of at least 1,100 RNA copies per ml and displays 70% diagnostic sensitivity. Merging this evidence with that of some other studies, including the recent technical note endorsed by the European Centre for Disease Prevention and Control, some practical consideration can be delineated for sample pooling, as summarized in this table. Therefore, we would recommend that this strategy shall only be used in areas with low disease prevalence, shall be used for population screening, but not for diagnosing SARS-CoV-2 infection in highly suspected or symptomatic cases. The pool shall be prepared by mixing upper respiratory specimen or saliva, but not extracted RNA. The locally used molecular assay must be accurately validated for being used for this purpose. The method based on RNA extraction are globally preferred. 5 seems to be the optimal individual sample size for each pool. The presence of interfering substances such as hemolysis or antiviral therapy must be ruled out. An accurate, preferably automatic traceability to the individual sample mixed within the pool must be assured. And finally, a second aliquot of individual clinical specimen shall be available for individual testing when the screening carried out on the pool turn out to be positive. Additional important indication are those concerning the assessment of the analytical performance of the molecular assays. It seems hence advisable that all laboratories should locally verify the claimed analytical performance of the molecular tests they are using, even if these are regulatory approved. A tentative list of all the parameters will be shown in the next slide, and also that all laboratories shall take part in a validated quality assurance programs whenever this will be possible. I will not go into many details for illustrating the contents of this table, which is available on the IFCC websites and defines the main analytical parameters recommended for clinical laboratories when verifying a regulatory approved commercial SARS-CoV-2 molecular assay. A paradigmatic example of potential analytical error in molecular testing of SARS-CoV-2 is described in this interesting report. Briefly, this short note emphasizes a case of oligonucleotide probes contamination during production caused by synthesis of full-length control oligonucleotides, which span from forward to reverse primers in concomitance with another probe order. This report thus highlights the importance of establishing truthful quality control assurance even within a context of reagent shortage and delay associated to huge volume of testing during a pandemic. As specifically concerned the specification for clinical performance verification of molecular tests for SARS-CoV-2, the IFCC recommends that laboratory should always verify the clinical performance claims of the manufacturers of molecular tests in a representative local population where the test will be used. When clinical samples are unavailable, and this specifically refers to repeat positives, positive or gold standard assays, or clinical criteria, contrived specimen shall be available and used. Finally, all clinical laboratories shall prefer follow the START guidelines when designing and reporting clinical performance studies. Some final consideration on appropriate interpretation of molecular test result before handing over to my colleagues. Positive test result applies when SARS-CoV-2 RNA has been detected so that the patient shall be considered presumptively infected, or when an active viral replication on the potential for viral transmission cannot be concluded. In such cases, clinical correlation with patient history is necessary along with obtaining other information. Negative test results apply when viral RNA has not been found in clinical samples or viral infection cannot be excluded, so that this test result shall not be used as unique basis for taking decisions. Negative test result must always be combined with clinical observation, patient history, and epidemiological information. Finally, retesting shall always be considered when infection is still suspected after considering other differential diagnosis. Molecular testing is used for hospital discharge, or some types of analytical inhibition are suspected. Indeterminate test result applies when test result cannot be interpreted, so that follow-up retesting may be recommended for generating a final result. I will then conclude my presentation with a brief consideration on the importance of using nucleic acid amplification tests that provide reliable quantitative rather than qualitative or semi-quantitative data. Besides the presence of multiple comorbidities, it has now been clearly demonstrated that the outcome of COVID-19 may be strongly influenced by the initial viral load. For example, this interesting Italian study found that the nasopharyngeal SARS-CoV-2 viral load measured upon hospital admission was a strong and independent predictor of death. In particular, in a fully adjusted multivariable analysis, cycle thresholds lower than 20 were found to be independently associated with a nearly fourfold higher risk of in-hospital mortality. Very similar data have been published in this other study, which confirmed the existence of a strong relationship between SARS-CoV-2 viral load in nasopharyngeal swab samples and COVID-19 mortality, with 7% increase in hazard for each log transformed copy per ml. The positive association between viral load in nasopharyngeal specimen of patients hospitalized for COVID-19 and hypoxemia on the right part of the slide and adverse clinical outcome in the left part of the slide has also been highlighted in this study. I will so conclude here my presentation. I am thankful for your attention